So yeah, I'm Mike Bottery. I'm a fellow here at Manchester, but I'm going to talk to you a bit about a piece of work I did back in my previous position at the University of York, where Microbes NG provided a lot of the sequencing, and the service was incredible. And I haven't been paid to say that. It was really, really good. So typically, we try and understand microbes. Classically, they're studied in isolation. So we try to understand their genotypes and their phenotypes, the dynamics of growth, generally in monocultures in the lab. And that's because it's quite tractable and easy to understand what's going on. But when you look at bacteria or any microbes in their natural environments, they are surrounded by a huge diversity of other microbes that may interact with them either competitively or have facilitative interactions and alter not only their phenotypes, the way that they grow, but their genotypes and their evolution as well. And this is particularly true within polymicrobial infections, where multiple different microbes infect the same site and contribute to the disease. And perhaps the best classical example of this is in cystic fibrosis. So cystic fibrosis is a genetic disorder which affects around 10,000 people in the UK and many more across the world. And it prevents the correct transport of chloride ions across the epithelial cells. And it means you get thick, sticky mucus building up in many organs, but particularly within the lungs, which allows bacteria to colonize the lungs when they shouldn't. And a main source of disease in people with cystic fibrosis is from a bacteria called Pseudomonas aeruginosa. So initially, these infections are intermittent and transient and can be cleared by antibiotic therapy, but eventually they evolve and adapt to life within the host and become chronic. These chronic infections cannot be cleared by conventional antibiotic treatment. So this is an evolutionary process within the host. And these bacteria evolve to the conditions of the CF lung. And they change many parts of their phenotype, from virulence to mobility, DNA repair, antibiotic resistance, biofilm production. A lot of traits change during this transition from this acute intermittent infections to these long-term chronic infections. And for some of these traits, it's kind of obvious what the selective drivers are. So for example, antibiotic resistance, well, the patients have been treated with huge amounts of antibiotics, both intravenously, through oral antibiotics and inhaled antibiotics. But for others, it's a little bit less clear. Why do these change their virulence factor production? Why do they alter the metabolism and other things? What are the selective drivers behind this chronic adaptation? So perhaps it's because within the lungs of these patients, there's not just one bacteria causing infection, but there's a huge diversity of different bacterial species in there. And some kind of typical co-occurring pathogens include Stenotrophomonas multifida, which is increasing in prevalence, or has been increasing in prevalence across Europe, and Staph aureus as well. So these are pathogens that can also infect the lung, but typically can be cleared through antibiotic treatment for some of them, but they co-reside along with Pseudomonas, this main pathogen. But to what extent these other pathogens drive the adaptation and evolution and phenotypes of Pseudomonas, it's very unclear. But it's not just the other species that are present, but it's the environment of the lung that could be the selective driver. So we all produce mucins, and these are the highly glycosylated molecules which cause the mucus to be kind of slippy and allow mucociliary clearance. But in people with cystic fibrosis, the chloride ions don't allow enough water into the mucus, so you get a too high concentration of mucins, and it creates this really thick, sticky, dehydrated mucus, which creates lots of spatial structure and actually allows the bacteria to form biofilms. So I wanted to ask, my main question was, to what extent do the in-species interactions and the CF environment, so through the addition of mucins, alter or drive the evolution and diversification of Pseudomonas aeruginosa during these polymicrobial infections of the CF lung? So I conducted the in vitro experimental evolution of Pseudomonas aeruginosa in the presence or absence of each of these competitors, so Staph aureus alone, or Stenotrophomonas multifida, or both. And then I also did this in the presence and the absence of mucins, so these mucins which create this thick and sticky media. And this was all done in media that replicates the nutritional environment of the CF lung, called synthetic CF media. I had 6 independent replicates per treatment, 8 evolving lines evolving treatments, so 48 cultures in total. And I evolved these for about 150 generations and asked 2 main questions. To what extent does Pseudomonas aeruginosa phenotypic adaptation and diversification is altered by each of these treatments, so the presence and absence of competition or the presence or absence of mucin? And can I detect the genotypic adaptation and diversification driving this phenotypic diversity? So first of all, phenotypic adaptation. I isolated 12 individual Pseudomonas isolates from each of my evolved populations and measured 8 different phenotypes, which are all associated or expected to change during chronic adaptation to the CF lung, including adherence, so how well do they form biofilms, some key virulence factor production, so pyrovernin, pyocyanin, protease, and elastase, and some growth dynamics as well, so growth rates, lag times, and max growth rates. So in total, so this is just I don't want to take anything away from this other than that I collected a ton of data. So there's around 14,000 phenotypic measurements from my evolved strains. And just looking at data like this, you can see things change compared to the ancestor, but it's hard to get a true picture. So we can look at things like multivariate analysis on this. So this is a multivariate analysis of diversification. To what extent do we see increased diversity in each of these phenotypic measures across this trait space? And across all of my treatments, we saw a big increase in diversity. So diversity is a measure of how wide of different ability to produce, say, these virulence factors, how wide the diversity we see in each of these populations. So we saw no real difference between each of the treatments. But when we dig down and look at the individual actual mesh phenotypic traits, we saw that the environments had big impacts on the diversification, specifically the presence of mucin. So the virulence factor production, virulence factors, so pyrovernin, pyocyanin, adherence, and elastase, and protease, all had much more diversity when you had the addition of mucin. So it means potentially mucin is driving independent evolution or within populations and between isolates from the same treatments. We also saw specific effects of competitor identity, particularly, say, in elastase, in adherence, and pyocyanin, where we saw different responses in trait diversification depending on the species that they're evolving with. So what about the actual phenotypes themselves? Did they evolve in specific directions within this multivariate trait space? So first of all, I'll explain these plots. So these are principal component plots which summarize in what direction was the traits moved across this multidimensional trait space. So the filled dots are the strains evolving in SCFM plus mucin, and the open dots are SCFM alone, so no mucin. So in the absence of competition, we saw a push towards higher pyocyanin production, slower growth rates, and higher adherence, away from the black dots, which are the ancestor. And this happened independent of whether mucin is there or not. But in the presence of competition with Staph aureus, we saw really quite different phenotypic trajectories, whether they were in presence of mucin or their absence. So in the presence of mucin, the phenotypes looked a lot more like when you evolved in the absence of competition. But in the absence of mucin, this presence of competition from Staph aureus drove the phenotype in a completely different direction. So suggesting that the presence of competition really can drive the phenotypic evolutionary trajectories. And this is also true with competition with Staphylococcus multifilia. Again, the presence of the mucins drove the phenotype towards a phenotype which more resembled the evolution in the absence of competition, potentially meaning that the presence of mucin and the spatial structure that it creates means that it limits the interactions between these. But in the absence of mucin, again, the phenotypic traits was much different and independent, potentially meaning that these interactions between the species was driving a specific phenotypic response. But interestingly, in the presence of both species, so this is Staphylococcus and Staph aureus, independent of whether mucin was there or not, all the traits were pushed in that kind of competitive, more virulent production direction. So potentially meaning that this impact of the spatial structure in the mucin was dependent upon the complexity of the interspecies competitions and the environments that were present. So can we detect any genotypes? This is where microbes NG came in and really helped. So we did 2 things. We wanted to ask, 1, does diversification change? And 2, does you see specific genotypic adaptation? So first of all, we extracted total DNA from the evolved populations and did deep sequencing across the entire population to understand the diversity of mutations that arose. But then I also I took those individual isolates, those 12 individual isolates, and individually isolated them as well, so we could get linkage between mutations and specifically link phenotypes to genotypes. So when we did the deep sequencing, it showed that most of the mutations were very low frequency within the population. So 85% occurred below 20% frequency, suggesting that most of the mutations that arose within this evolution experiment were potentially under soft selection or arose due to genetic drift or by piggybacking on more beneficial mutations within the population. But the other 15%, so 49 mutations, which were across 23 different loci, occurred at above 20% frequency and were overrepresented by non-synonymous mutations, really suggesting there's strong positive selection for these mutations within the evolution experiment. So what were these? So these rings just represent bacterial genomes, and the dots show where the mutations occurred. And the vast majority of these parallel mutations, which are at high frequency, were in virulence factor production regulators, such as VQSM, PQSR genes, as well as the TADZ, adherence genes, and also alginate production as well. So these are mutations which potentially are driving these changes in the virulence factor phenotypes. So we wanted to dig down and ask, right, what exactly are these mutations, effect are these mutations having? And so this is a whole population genomics. We can't really tell linkage, so we sequenced individual isolates to ask what was going on. And when we did, we noticed something really interesting. In this VQSM, which is an operon, which is a master regulator of virulence factor production, in the absence of mucin and the absence of competition, we saw really large deletions within this operon, which can profoundly alter the virulence factor production. But we never saw mutations or deletions in any of the competitions with other species in the absence of mucin, showing that the virulence factor production controlled by this operon was potentially essential to maintain this cooperative virulent phenotype in the presence of competition. But in the presence of mucin, we saw these deletions across the operon in all of the no competitor treatments and the single species competitor treatments as well. Again, suggesting that the presence of mucin was potentially isolating the competitive impact of these individual strains. But interestingly, we never saw deletions when we had both strains together, this more complex multi-species population. We saw a couple of individual SNPs, non-synonymous SNPs within VQSM, but never the deletion and never the loss of function. So to determine what effect this was having on the phenotype, we could isolate those genotyped strains and measure the phenotypes again. And we see that, so the two that we really want to focus on is the VQSM, so this is where you saw the SNPs, and this is the deletions within that operon. And we see quite different phenotypic trajectories between the two. For example, in elastase production is completely halted when you delete this have deletions in this operon, but the SNPs actually elevate this trait. And again, we see differences in things like pyoverdin production, pyocyanin production as well. But it also has big effects on growth rates as well, potentially meaning that the growth rates in these strains was quite important for to maintain competitive phenotypes. So back to the original question. To what extent does interspecies competition and the CF environments, the presence of mucins, or drive the evolution and diversification of Pseudomonas aeruginosa during polymicrobial infections? So we show that the diversification on a whole isn't really affected, but it is affected by individual traits, But the interspecies competition led to the evolution of a distinct phenotypic trajectory, only absence of mucin. But in the presence of mucin, it played a critical role interacting with the presence of competitors, which meant when you had a multi-species environment, the phenotypes were driven to a more competitive phenotype. So this might help explain why during chronic infection of the lung, Pseudomonas generally is thought to lose virulence factor production, slow down growth rates, but you pretty much always can still isolate high virulent subpopulations from the lung. And this is potentially driven by the competitive interactions that are present. But you've got to remember the lung is a very spatially heterogeneous and complex environment, so there's still quite a lot of understanding that we need to get. So with that, I'd like to thank everyone for your attention, Microbes NG for providing the opportunity to speak, but also my mentors at York, so Ville and John were fundamental in putting this together. I think Ville is somewhere if you want to catch up with them. And also my collaborators at Technical University of Denmark. So with that, thank you very much, and no questions, I think.